going to share it on the YouTube as well. So it's going to be on YouTube and it's going to be on our website as well. So those that want to go back to the lecture, you would have the opportunity to do so. But you need to attend the meeting to be able to get a certificate. So we are glad you're here. Yeah. So Dr. Sarah Osafo has been a colleague of mine. She did her PhD at the University of Ghana Dental School, and her major was in biomedical materials, and he did coatings, HEA coatings, and graphene coatings for osseointegration. And osseointegration is an area where you try to accelerate the attachment of cells on the surface of various implants. And so she's done some dental work, she's done some metal, titanium alloys work, and her work is published at very high impact factor journals as well. So we are very glad to have her around and to be with us. So, Dr. Sarah, over to you when you're ready. Just signal us and you can start the lecture.
Okay. Thank you very much, Dr. Tabiri, for the introduction. And I'd like to appreciate the young African scientists for an opportunity to share with all of us this afternoon, morning, evening. So good morning, afternoon, evening to all of us from wherever we are connecting from. Like he has said, today I'm going to share with us let me test my screen. Okay. I'm going to be sharing with us on the next generation biomaterials, highlight some opportunities and some challenges. And Dr. Tabiri has already introduced me. I like to say that I am made in Ghana, Africa, and connected to the world. I had my BSc and MSc in Africa, up to PhD in Africa, and had opportunities to access certain labs. And normally I like to highlight on this because sometimes based on our backgrounds, our environments, we tend to see challenges, but then with the little we have, when we're able to make do with it, we are able to contribute to groundbreaking discoveries in science. So that is me, made in Africa and connected to the world. And this is the Young African Scientist. So you're all welcome to today's lecture series, which is going to take us through how we understand biomaterials and how to contribute to advancing it to the next generation as it is an evolving field. And so for today's journey, I will take us through what biomaterials are, why it even matters for us to talk about it, the different types available, its application in the healthcare, and a case study on its evolution, some opportunities, challenges, and some future directions. So without wasting much time, I'll get right into it. So when we talk about biomaterials, before now, when you Google biomaterial, you look into textbook, one of the key things you find is that it is a non-viable material. That means that it is not a living thing, but something made out of materials that do not live. So that means they cannot grow on their own. But these are used for medical devices, numerous applications, and in their usage, it's always intended to interact with biological systems. And its interaction is very key to its evolution, how biomaterials has advanced. You can find biomaterials existing in natural forms or synthetic forms, and they are used in a very widespread application in the healthcare field. So from prosthetics, diagnostics, therapeutics, storage, and a whole lot in the medical and dentistry field. Now, like I said, when you look at the definition, how it interacts with the biological system is what has led to different generations of biomaterial science. Before, when we talked about biomaterial science, we just needed materials that will be used in a living environment, and we just needed it to do its job, and that's it. So to be able to exist in the physiological environment without causing problem. And that is the bio-inert age, where materials were just supposed to be tolerated. And by that alone, we measured the material to be successful. An example, those times, common materials that were used were stainless steel, PMMA, cobalt, chrome. Then it got to a point where, yes, we need materials to be tolerated in the body without adverse effects. But at some point in time, beyond the material just being tolerated, we needed them to be bioactive. That means that we needed them to form some strong bonds or interaction with their surrounding tissues or hosts that they are implanted into. So that led to the research and development into materials that can do that kind of interaction. And then also sometimes when materials have been used inside the physiological environment, we mostly after usage, so if a material is say used as an implant and it finished doing its job, we need to go in again through another surgery and remove it. And that is cost, that is inconvenient to the patient. So as the development of bioactive material existed, then came also resorbable material. So I'm sure for our women here, when we go through CS section, before the material that we're using suturing, after some time it will be removed. But then with with the evolution, there is resorbable materials whereby after the suturing, the material just melts into the body, so you don't have to remove it again. And then from the 2000s, we got to a stage where we want to be able to instruct materials as to what to do inside the body. So beyond tolerating, being active and all, we want to be able to instruct some growth factors on the materials. So instead of cells just interacting, we should be able to design materials in such a way that we're able to instruct cells with some cues and they execute that whilst inside the body. And then 2020 onwards, we are getting towards living and adaptive materials. So materials that would be able to behave more like live, materials that can live. So we are moving away from the non-viable material such that we can design an entire heart, which will be able to, instead of patients waiting for transplant when they need any organ, we are getting to where we design materials that can sort of live. So that is where biomaterials is evolving from based on its simple interaction within biological systems. Now, why it matters? I normally answer this question based on statistics and ongoing discussions in diverse fields. So as humans, we all gathered here want some good quality life. We wish we would live forever young, and as we age, we will not be having knee pains. You know, when you're a child, once you lose a tooth, a new one would grow. It gets to a point you lose a tooth and another one is not going to come because nature is structured in such a way that at some point certain things doesn't happen. But then with biomaterials, we are able to cause some changes so that we are able to have things to give us the quality of life that we desire. With aging, we tend to have bone defects and all of that. Even sometimes just trauma. You get hurt maybe in an accident and all, and you lose some parts of the body. And biomaterial science makes it possible for certain tissues, for certain parts to be replaced. So biomaterial science is a field that is very important and it matters to every one of us here because if we lose a tooth and we want to say have our beautiful smile, and by virtue of biomaterial science, we are able to have an implant, for an example. If our hip is fractured and based on our age, the tissues or things cannot repair it, biomaterials can help for us to get a hip implant and all of that. And then one thing that most people we don't like to hear is cancer. which is also something that affects every race and every one, male or female. Now, with cancer, so many advances are being made in dealing with cancer. There's a chemo, there is a radiotherapy, and all of that. Now, how does materials come into play? In cancer treatment, the doses tend to be very, very high. And so when patients are receiving cancer treatment, oftentimes it comes with other side effects because as they are treating the cancer cells, it's also sometimes killing some healthy cells. So with biomaterials, there has been the development of what we call targeted drug delivery. And that means that, say, we tried to treat breast cancer through surgery. After the tumor is removed, we still need some medication just so that it doesn't regrow. So through biomaterial advances, we're able to design drug delivery devices which can hold some medication and implant it into that space. And it will be designed in such a way that over time, it gives medicine to that location. So all this makes biomaterial a really important field to all of us gathered here. Now, just like the general material science, we have different classes of biomaterials. For each of the classes, you would have different groups of materials in there, and every class will come with its advantages and some sort of disadvantages for you to deal with. Now, when we take metals, for example, metals, some examples of metallic materials that are used in biological applications, we have titanium alloy, we have cobalt chrome, we have stainless steel, magnesium, and zinc, and more. What's the advantage of these is that, or what it is buying you is strength and fatigue life. So when you are using metals, it's able to withstand load. It's able to have a long fatigue life. So that means that you can use these in applications where there is a lot of load. So for instance, for a hip implant, for dental implants, as we are chewing, as we are walking, we are applying stresses and load to those areas. So metals is able to give us that. Now, as it's giving us all of that, what it also causes is sometimes some stiffness or mismatch. So for instance, stainless steel or titanium is used as an implant, dental implant, an orthopedic implant, or whatever. It has its own modulus, which tend to be very, very high than that of the normal cortical bone. So there is a mismatch. So to picture it, say you have lost a tooth, and then we put a metal there to be able to carry your crown. Because the metal is very, very strong, it carries all the load from the chewing, preventing the surrounding bone existing there not to also receive some of the load. And that alone can cause what we term as stress shielding in material. So the other surrounding material tissues are not able to absorb some of the load, and so that can also cause failure in that material application. Then we have ceramics, which also includes alumina, zirconia, hydroxyapatite, bioglass. And one advantage of these classes is that it presents us with a very good wear resistance, because when you take metals, they are hard materials, and so when you scratch, it's able to resist it and becomes wear resistance. And so for some applications, we wouldn't want materials wearing into the physiological environment. So our best bet would be to go with this class of materials. Now, and it's also bioactive. That means that when we looked at the evolution where we are moving from bio-inert to bioactive, ceramic is one class of material that gives us that advantage. So it's able to bring us that kind of interaction between the material and then the surrounding tissue. It also comes with its own challenges, which is brittle, and it's very difficult to shape. And as it is giving us the hardness and all, it's also not that tough. Toughness is when an impact is applied to the material, its ability to absorb it and not fracture. So when you scratch a ceramic, it might withstand it, but maybe when you impact with a load, it can break or it can fracture. So that's ceramic. Then we have polymer, which also has different material in them. Ultra high molecular weights, polyethylene. We have PEEK, PLA, PCl. All these are types of polymers. And one advantage of polymers, which also makes it a very, very desirable biomaterial, is that it is tunable. So when you take polymers, take your mind to polythene bag, nylon, and all of those. They are made of long chains of network. And so you are able to do so many things with it without altering its chemical properties. And so in processing, we're able to tune it into what we need it to do. So if we needed some type of mechanical property, we can make adjustment and still not alter its chemical stability. So that's the advantage of polymers. And it's also resorbable. So where we need materials that after performing a function, we still don't need it in the location of use, we would go for polymers because it can resorb over a period of time. Now, its own disadvantage is creep. After a long time, under its own weight, the material can fail. Then it can also cause acid degradation and then wear debris into the sites where the materials are used. Now, these three are the major classes of materials. Now, in material science and design, sometimes a material possesses a very good quality that we need, but then you will see that it is missing in another, just as we have seen with what it buys you and then what it costs you. So what material scientists would normally do is that, okay, we need metal, but then because of its stiffness, it won't do so well in a particular application. So we can bring another material to give us that property. So that is composite materials, which you can reinforce PEEK, you can reinforce other metallic materials with different, any of the classes that has been mentioned. And so another advantage for this is that it's able to, because we are able to also make composite ourselves, we are able to design properties that match tissues or the biological systems that we are going to use them for. Then its advantage is that we sometimes have interface failure. Most often than not, because we are using two materials, sometimes we want them to bond and all of that. And based on the strength of that, it could fail under that. And then they also have some complex qualifications that makes it very difficult to come by. And then the last class, which is now a new class of biomaterial, is the living and the hybrid, where we are getting to materials that are made out of biological products. Like we can make a material with cells involved in it. So using printing, 3D printing and all, we can print materials that behave like living systems. And those materials are able to adapt to the environment. They self-repair just like the way nature made it. Now with this one, the shelf life as it is now, we are not able to design those materials to last a very long time. And it's also very, very cost intensive. And there are no, because it's a new phase, regulatory is not that standardized yet. So with all these classes of material, you ask yourself, so in biomaterial application, what then becomes an ideal biomaterial for us to use? And I believe from the previous slide, we have our answer. We normally do not have what an ideal biomaterial is, but you can have an ideal fit based on the intended function that you want a material to perform for you. So I looked up different materials in different classes and some relative suitability in terms of strength, stiffness, degradability, bioactivity, and imaging. And if you look at this slide, you would see that there are different materials and not a single one of them was the best throughout. So this is to tell us that you get one material giving you the best strength. Then when it comes to stiffness, it might not give you the best. When it comes to degradability, that might not be the best and all of that. So in biomaterial and what we term to be ideal, when we are looking at an ideal biomaterial, we are looking at fit, we are looking at functionality. So for a particular application, what do you intend for the material to be? So based on that, you go ahead and choose materials that would give you that exact kind of properties that would be suitable for you. Now, with a bit of understanding of what biomaterials are, I want us to have a fair idea of how big this field is. So biomaterials is not a niche kind of field. It's something that exists in the healthcare ecosystem. So in the healthcare, different areas of healthcare area, biomaterials is used. And when you look at some research data on the market, there is a wide variation on what is reported. So you see that there is a range of about $45 billion to $200 billion. That is how big this field is. So you'd wonder, why is there this variation? So when you take any statistics, what you should ask yourself is, what was the factor that was being measured? So biomaterials is a wide field, and sometimes people are measuring it from different perspectives. It could be measuring it with the raw materials. It could be measuring with medical devices, with the application, healthcare products. So all these differences lead to the different reports that the same field is getting how big the field is. But one thing that is not in dispute is the clinical need. Regardless of how big or how small this field is being represented, there is a clinical need for biomaterials. Every field of the healthcare ecosystem needs biomaterial for implants, for auxiliary materials, a whole lot. We need biomaterial. So with this understanding, where is the field going? Where is the materials field going? Now, when we understand the materials, their applications, like we have mentioned, the advantages and their limitation, the next thing to ask is, where is it going? We saw how it started from bio-inert materials to living materials. So where we are no longer just replacing tissues, but we are getting to the point where we want to make these materials active. We want to make it instructive and to be able to adapt. That means to regenerate. So when you lose a part of the body, biomaterials is getting to the point where let's say one common example that I'll be using because of my field is I work in the dental school. And so when when patients lose their tooth, now you are able to, based on aging, which I mentioned as part of the statistics why we need biomaterials, when you get older, the way the body is able to self-heal itself reduces. So as we age, if we needed an implant, maybe because of your age, the bone quality there will not be favorable. But then we can design materials to be able to fill that particular space and make it adaptive to regenerate, more like grow back the bone that you have lost so that an implant can be done. Now, when we look at this thing, I'm sure it looks like something that might seem very challenging to do, which is true. It is challenging to do. But even in environments like Africa, it is still very, very possible to contribute to where this field is going. In our small labs and all, we can come up with material design. And in answering where it is going from the materials or engineering point of view, one key thing is failure. At every stage of advancement, so we make implants and they are just interacting or being tolerated in the body. And because of that same opportunity that we had, as the material is being used, we find that it tends to fail in some aspects. Then it gives us an opportunity to do some more modification or advancement on it. So for instance, in the medical field, when we use materials as, say, implants, sometimes there could be infections, there could be growth of biofilms on the material, and all of these tend to hinder the success of the implant. So if we are to implant, let's say, a metal, and it's supposed to last a length of time, because of infection, bacteria, and biofilm formation, that material will not be able to be very, very successful as we want it. Then the stress shielding that I explained, we have used, let's say, a metal as a load-bearing implant, and because of its mismatch in terms of the stiffness, it is shielding the other part of the other part of the bone from absorbing some of the load. And so with that, the material tends to fail. So the implant that is maybe supposed to last for maybe 20 years or so might not be able to last that length of time, causing patients to in no time come back to the hospital for revisions and all of that. Then we also have fibrosis and foreign body response. So when we implant materials into the body, depending on the kind of application, we sometimes want it to just interact without any interface, and that is also integration. But some materials are not able to do that because of its bioinertness. And so it ends up with material forming like fibrosis, that it forms a scar before it interacts with the body. And that also makes the material to fail. We also have wear, debris, and loosening. So all these are failures that can occur in our use of biomaterial. So when we identify this failure, then it sets the research agenda for us. So why is the material failing in this regard? What can be done? So as we observe this failure, then we are able to come up with new innovations. And if you look at the different evolution of biomaterials that I mentioned, we don't just leave one generation of material just because we are advancing. One material can be advanced to give us another generation of material. So as we use them, so you have the opportunity of using these materials in clinically, then we observe the challenges, which is the failure. Then we come up with new innovations to advance the field to where we want it to get to. Okay. So with the evolution and how the agenda for advancing this, every limitation we find with one generation of material becomes the opportunity to advance it. So as we can see here, we have an implant. We have an implant material here, maybe a knee implant, and it could be made of steel, some type of metal. And then these are being tolerated very well by the body. But it gets to a point where it take long for integration to occur. So we are now looking for new materials that can bond, that can degrade, that can integrate based on the application. And so for instance, ceramic material are a class of material that does really well in this regard. Then to the instructive where we tell cells what to do. So for instance, in design of materials, let's say ceramic materials, we are able to design ceramic materials in such a way that it's able to always alter the surfaces of some materials such that it instructs cells on how to behave as soon as they come into contact with them. And it behaves exactly that way, leading to that kind of functionality we need a material to do. And then with the living and adaptive, now we are getting to a place where we have organoids. So it will get to a time where when we lose some of our organs, we will be privileged to have materials being done from live cells and tissues to be able to use as implantable material without waiting for donors to give us this. And that will be a very huge advancement in the field. So we can see that biomaterials have evolved from all of these stages, from opportunities of having them, the challenges that we encounter once the materials are in use. And from all of these, we get to move the biomaterials towards different generation or the next generation of biomaterial science. So I'm going to use a case study of implants for how biomaterials has evolved. And like I mentioned, sometimes in our little labs as researchers, when we are doing experiments, I see that we mostly would report what worked, but not much of what didn't work. But all of that advances a field. So in the field of biomaterials, implants were already being used, and Brainmark was studying bone microcirculation in rabbits. So he was using titanium as a carrier. So he needed a microscope to get to a location in the rabbit, and the carrier was the titanium. And after some time, after the experiment was done, he realized that it was now very difficult to pull out the titanium. So then the term osseointegration came by. So that meant that the metal that was meant to go in and come out had gone to fuse with the surrounding tissues. Now, when you look at when that was done, that was 1952. And then the only time a clinical test was done was in 1965. So in advancing biomaterial, sometimes it takes time because of different things that needs to be done for us to say that this material is ready to be tested in animals before we get to human trials. So after this discovery was made, and we came by osseointegration in 1965, it was then tested clinically to becoming the first clinical implant that was done. So with all of this, it has led to different ways of improving the state of one material to another based on its functionality that is required. Now, the question is, with all these advances, are we able to even engineer metals more to even do some better work for? And better here is not necessarily good. Better is functionality, what we need the material to do. For some material, better may mean that it should just fuse. Some material, better may mean that after some time it should degrade without we having to remove it from the body. So that is with how certain advances have been made. So I believe that in our various labs, as we do our experiments and all, we are able to come up with some of these things. So using dental implant as a very practical example and tying it to osseointegration that was discovered in Brainmark's lab. So titanium, which is like the gold material in dentistry, it is such that it has the mechanical strength, it is biocompatible, it has some wear resistance property and all of that. So whenever an implant needs to be done, this material will be implanted in the jawbone. And after some time, when it has been able to heal, which is the osseointegration phase, an abutment will be implanted on it. And then you have your crown. Now, it's being bio-inert. Now, we want to take this bio-inert to a different generation of material, which is bioactive. So what can be done? And in doing some of these advances, we can use some core material design to be able to achieve that. So for instance, every material would have a structure, and based on the structure of the material, it also possesses certain properties which can give us some functionality that we require. So when you look at this image, it is an image where the left part is the natural bone to the tooth root and then an implanted device. So this implanted device is supposed to behave just like the tooth root is going to be behaving, but definitely in terms of its chemistry, in terms of its modulus, there are some differences. So what can we do? So we look at what can we engineer? Maybe we can do something to alter the chemistry of the material. We can do something to alter the face. And as we think about the structure and the requirement, then we now start looking at what materials are available, what material is desirable. So for instance, hydroxyapatite and some graphene some materials that, for instance, hydroxyapatite is the inorganic material in the bone. So when you take any natural bone, the inorganic component is hydroxyapatite. And you can find them in animal bone. You can find them calcium carbonate in eggshells, and use these materials, capitalize on their structure, and do some synthesis to get the desirable properties that you require out of them. So once you're able to successfully do that, then you get the functionality that you desire. So what is the biological experience? When a material is implanted, we need it to interact with the proteins around the implanted surface. We need it to attach with the cells. We need it to spread. We need it to mineralize, and we need also integration to be very, very stable for the material to be able to do its function. So as you can see, we have a system. We do some surface modification on it by altering the structural changes to give us the properties we require to be able to get the biological response that we desire. So all these are steps through which we are able to take existing materials to the next generation that is required. So in the steps that I showed, we can use surface modification as a tool to drive in the evolution of biomaterial science. So because of titanium example I've given, which is an inert material, we want to do different surface modification to ensure that it has a structure that the normal bone does. So different approaches have been used. Some people use anodic oxidation, laser radiation, etching, grit blasting, sandblasting, and then coating methods. So when you take an implant material that we need it to become bioactive, what we need is that the material should be such that when it goes in contact with the cells or tissues, it should be favorable. The cells should see them as one of them so that it will allow the attachment to occur. And normally, the cells, just like when you are working on a very smooth surface, you might slip and fall. So we need some roughness so that the cells are able to hold on to. So that is with some of the surface modification, what we normally intend to achieve. We want the material to have some kind of topography, which is not smooth, but then some extent of roughness. And all these approaches are done to ensure that. And one that I will zero in on is the coating method, where ceramic materials are coated onto metals available to move them from being bioinert to being bioactive. So one approach that I used for a surface modification approach was coating a metallic material, titanium, with hydroxyapatite in a very simple approach. So in any lab that you can have a furnace, this can be done by synthesizing your hydroxyapatite. In this case, I synthesized them from bovine bone. So through processing, some processing techniques, you're able to get hydroxyapatite and then pack it around the metallic material, then heat it to high temperatures. So through diffusion, this metallic is going to coat this bioinert material and then make it to become bioactive. Now, as we are thinking of advancing the field, when all of these have been done, we need to be able to characterize the materials to see whether the structure and the properties that we were hoping to see has actually taken place. So for instance, the material that I coated, this is it, and this is how you see it with the physical eyes, but with some advanced characterization, we're able to really see the structure. So this thing that we just see as some dark material, upon characterization, it gives us the structure of the material. So for instance, I mentioned that for implant material, we needed to have some topographies and then some chemistry. So our characterization in the lab is able to tell us that, okay, indeed, this material has grown some nanorods, which tends to make it that desirable topography. And then also when we check the elemental properties through EDS, we're able to see calcium and phosphorus on it. So all these are ways where, as we attempt to advance material, we are able to, utilize on these techniques to do so. Now, sometimes you may be constrained in terms of resources based on where you are. And one thing that I would say is whatever you find, the little resource that you have available to you, when you do it repeatedly and kind of perfect it, it gives you the ability to collaborate. So say you have a lab, you're able to do all these things. Maybe the simplest you can do is just the coating, but you don't have the means to characterize them. Once you understand the science, you understand the chemistry, the physics, and all that is going on, it gives you leverage to collaborate with people in advanced labs to be able to characterize your material, to be able to do some more advanced research on a material, to be able to come by a product or to advance a material from one phase to the other. So that happens to be sometimes constraint that especially we in Africa will face. Maybe in your school there might not be SEM and you need to know the morphology of a material. So just whatever it's available to you, make sure that you have that good understanding. And as we come together like the young African scientists, we have the opportunity to network with people and take our research to the next level. So with that modification that I shared with us, this is just to show one improvement that it did. So normally when titanium is used as an implant, it takes about days for the cells to begin to interact with the metal. But because we successfully moved it to being bioactive by changing the chemistry, now the chemistry of the metal is not just titanium, but it has calcium phosphate. It made it chemically suitable for the location of implantation. So, so this is just a cell surface interaction where a material has been coated. We want to see what has really happened. So we put cells on it to observe the interaction. So normally cells are spherical in nature at the early stages, just the nucleus and the cytoskeleton. So we observed it within a number of time points. So our observation was that at the early stages, all the so we have titanium, and then we have titanium that we coated with the hydroxyapatite I mentioned from different sources, bovine and an eggshell. And for the coated ones, what we saw was that based on the structure and the property that we're able to introduce onto it, at the initial stages, they all had a spherical nature. But then in cell attachment and growth, there is some features of the cell, which is the phylopodium and lamellipodium, which normally will start to extrude and cause the cells to begin to move. And we saw that as early as 15 minutes. Then at 30 minutes, we could see that for the titanium, the anneal titanium was used as a control. So that was still pretty much spherical. But then the coated one started losing their spherical shape and started spreading on the substrate. So with this, we could see that indeed we have been able to take a material, a biomaterial from just being bio-inert to being bioactive by coating methods and being able to characterize them to see their properties that we come by. So we could see that from challenges, we are able to have opportunities to work with materials and take them to the different phases and functionalities that are desirable. So when you look at this image, a titanium metal that is bio-inert, based on our coating method, we are able to introduce this roughness onto them, whereby we have these nanorods that helps the material to function very well as an implant material, thereby reducing the chances of failure. So if you see the dentist and you a material that has been able to be advanced like this is used for you. For the patient, it is convenience, it is saving them money and pain, because if any time you have an implant and you need to do revision beyond the cost, that means your mouth being worked on all the time. So that is one advantage of being able to use basic understanding in material, that is structure, the properties, and then the functionality to advance material to new or different generations that we require. Now, when we have found all of these, it doesn't end there. Promising material is only the beginning. So when you successfully, because biomaterials are mostly going to be used in contact with humans. Some of them are auxiliary materials, so that one not much is required. But for the ones that will be inside the body, we need to ensure that it is safe and it doesn't cause any harm to the body. So after being able to do cell works, another thing is to be able to do animal studies where a material that has proved to be good or proved to be functional based on our characterization would now have to go through animal studies where we implant them in animals to observe certain reactions. After that, there'll be clinical trials, and if everything is okay, then the material will be okay to be used in the real world application. So these are some of the stages where materials go through before they get onto the market. Now, I've said a lot. And in every field now, AI is becoming very, very crucial. And so for every field, with biomaterial not being an exception, as we advance in our experimental and all, we need to also look at the role of AI, what it is playing, the role AI is playing in all of these things. And one thing that I came by, as I said, some of these bodies that regulate the field, is that FDA, for example, published a roadmap where they want to reduce animal testing and have more of computational and AI replacement. And NIH also wants to stop funding proposals that rely solely on animal experiment. That means that even if we are using animal experiment, we want to get to the point where trial and error is reduced. So artificial intelligence comes in very handy to help us to come close to what is true before we go into testing things on animals, because there are people who are concerned about animals and we don't want to disturb animals for no cause. So when you look at AI in biomaterial science, it's helping in designing. So with all the example that I just showed, sometimes you have to be in the lab constantly trying things to ensure that you come up with a good design. But from a materials perspective, once you understand a material structure, you know how to alter the properties, you are always able to tell what the function will be. So AI helps with existing results that are available, material properties, material structure, to be able to design without the burden of in vitro studies and all of that. Now, it doesn't also just come easy like that. There is always some limitation, especially when a field is new and advancing, the burden of validation. So no matter what you do, you would always have to also validate some of the findings that AI helps you to come up with. AI also helps us to predict. So I understand that metals have this property. I know the property of hydroxyapatite. I want to bring the two together instead of going to the lab, trying different temperatures and all of that. And I can rely on some existing data and predict that, okay, when I heat like the coating I did with some data, you can I'm not an expert in machine learning and AI, but then the little I know, you can do some prediction and say that, okay, when you heat a material at this temperature, you are able to get this functionality. And looking at AI in biomaterial science, one challenge that I found was small data. And I looked at it in a way that in every challenge, there is also an opportunity. So although there's a challenge with small data, because biomaterial science experiments tend to be very, very costly. So getting large volumes of data to do deep machine learning can be very difficult. So although it's advancing, instead of waiting for the field to grow, there is an opportunity to, come up with small data methods that will help this field on its own, so that there will be tools that can work with small data and make impact. So from this, a colleague of mine who is into machine learning and AI, I try to get her point of view with small data and what can be done, and using my research as a case study. So even with small data, what kind of machine learning can be constrained with biomaterials research, as I've mentioned, because of large data that is mostly needed. That's a challenge. So what can be done? The opportunity is that as we are able to, so before we go into anything, we have that understanding. So the understanding of the science behind everything is very key, and which we will never be constrained with. We would always have an understanding of material processes, material structure, and the relationships that exist. So based on that, it helps us to prioritize our experiments and all to be able to work with small data that exists. By so doing, a small data can be used to help in advancing a field, as now it has become a part of biomaterial, just like any field. AI can never be taken out of the conversation on any topic nowadays. So with regardless of the small data that we might have, we can still make do with it, although we may have small batches that we are working with, but a deeper understanding of the science that we are working with, that data can always make a big impact for us. So as I did some of these experiments, even with the small data in quote available. Work is being done on it to be able to make some predictions to advance the work to its next stage. So, bringing everything together. So, in taking biomaterials from the bio-inert to the next generation, which is bioactive, which is the living biomaterials, what normally presents as a clinical challenge is a real issue. And through biomaterials, we're able to identify problems, and these problems serve as our opportunity to address them through materials innovation by engineering them, by altering their properties to get something through structure, property, and functional approach. So one method that's material scientists are able to use to alter whatever functionality they desire is structure and property, which is very, very key. And so in my own research, I have used this approach to move a biomaterial from an inert to a bioactive stage by enhancing its osseointegration, some antibacterial property, improve their tribological property, get some mechanistic understanding to take it to the next level. Now, the translational pathways, after any successful experimental approaches to enhance or modify biomaterials, it needs to go through animal studies, clinical trials, and then we begin to make real world impact. And in all of these things, the role of AI and machine learning tends to be very, very important. So all these are opportunities that biomaterials, physics, chemistry, computer science, data science can see opportunities in biomaterials and help in advancing the field. So, if you don't remember anything from this lecture, some of the key things I want you to take away with is that there is no ideal biomaterial, and that makes the field an interesting one to contribute to. And whenever you see a failure, it is your drive for innovation. Like they say, necessity is the mother of all invention. So when we see materials fail in certain regards, it's our opportunity to contribute to making them better, engineering interfaces, and knowing that translation raises the evidence burden. So there is that opportunity you can tap into. And AI can make every experiment count, synthesizing all experimental data and coming up with predictive tools to advance any field is very key. Thank you so much for your attention. And once again, I would like to acknowledge the young African scientists for an opportunity to share with us. And also, I'd like to acknowledge the University of Ghana Dental School, SUNY Polytechnic, for access to the labs, and my mentor, Prof. Wole Soboyejo, and all my research collaborators so much for the collaborations that we do together to come up with some of these findings. Thank you very much for your attention.
Yeah, thank you very much for the lecture as well. It's been very interesting. And I work with you a lot, but I've learned more today. In the past 2 weeks, as we have been talking, you've not been saying these things to us. So this is a very nice presentation. Thank you very much. Now we want to open up 15 minutes for questions, and Dr. Sarah is here, so she'll be willing to provide some answers for you. My best part of the presentation is almost in the early stages when you talk about the various stages of biomedical materials, the new advances, what we want now. So if you can go to that slide and share a bit more light, that would be helpful. And as she does that, please, if you have a question, you can leave it in the chat. If you
Is it this one?
No, there's one more that uh, yes, no, keep going. I'll tell you
Okay.
which one it is. We're going back. I had that one. No, that one.
Okay.
Go back. Okay, yeah, it's I think it starts from it, but go back one more. Yes. So the graph under the where you have the passive, active, and those stuff. Yeah.
Oh, okay, okay,
Yeah, adaptive. Yeah.
okay. So with this, as I explained earlier, so before materials were not really required to be active, like we just place it there. And so when you look at some old textbooks, when we talk about biomaterials, the focus is that we don't want it to elicit any adverse effects. We want it to be safe, and that's it. And that's with the passive. Now, with driving it to being active, it's not just about not eliciting adverse effects, but rather coming up with interactions. So that interaction should be such that it's able to communicate with its surrounding and not just be there passively. Okay, so in fields like drug delivery, implants, when we implant materials in the body, say as the at the breast side, the tooth, and all of that, we need there to be an interaction. So if the material is passive, it will be very, very difficult for that implant to be successful, to even have that drug delivery taking place. So the material should be such that it has the ability to communicate with cells and tissues around there. So that's moving it from passive to active. And in that same quest, so how we are able to do that? So now we come up with materials where we introduce certain proteins onto them. So say I'm doing a coating with graphene with hydroxyapatite. I could also coat some, extracted extracellular matrix. In that way, as soon as the material, that is us designing the material to be able to instruct. So when the cell gets there, it's able to, um, in simple terms, instruct the cells on what to do. And before, especially with implants, we used to look at implant as just putting the material there. But now, where it's going, we see that how even osseointegration occurs is that the body tends to release cells that sees the implant as a foreign body, a foreign material. So then the white blood cells are normally released to kind of attack it. And that is how come it's able to form that bond around it. So that is instructing materials on how to behave and then eventually to be able to be adaptive. So the metal is going to be in the body and it has to be able to adapt with that particular location and function as the natural tissue or whatever we have replaced a material, an implant with would behave. I hope I
All right.
have.
Yeah, 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 yeah. It's very understanding, and that I think shapes the future of the field. And that means that Africans we have a part to play. So,
Yeah.
what's the level of instrumentation you think can bring this whole thing into technology ready setups?
Mm.
Uh,
So, with instrumentations, the basics is that when you have done all these experiments, we always need to know what we have done. So materials will need to be characterized and we could have different focus and that will determine the instrumentations that are needed. And that's why I introduced a bit of collaboration and network because sometimes most of our African universities, you hardly would find some of these advanced tools for characterizing our materials. But then the understanding of the science itself gives you access to places where you can collaborate and then continue with your work so that we are not lagging behind. Yes.
all right. So we are still, I still see some of us have not registered. Please, if you need a certificate for this lecture, or lectures have been considered as training lectures. So you want to show this certificate in biomaterials if you're applying for a scholarship, if you're applying for a job or anything for people to know you have this basic understanding into biomaterials. So please make sure you use the link that I've been posting, and I'll post it once more. Just go to the link and fill your information for us, and we'll register you in our system and we'll send you the certificate when it is ready. All right. Any question for Dr. Sarah before she goes? Any question? Anyone has a question for her? Do we have some people that are already doing biomaterials in the group? We would want you to say a few words. Uh, Dr. Sarah, I think there are some questions in the chat for you, if you can take them for us.
Okay.
In terms of drug
Okay.
delivery, especially when working with inorganic materials, is there any progress in removal of these nanomaterials from the human body to avoid bioaccumulation?
Okay, so when it comes to the removal, that is what has been the order of the day. But the progress is now trying to, even if it's an inorganic material, design it in such a way that after the period where we need the material to be used, it will be able to resolve without, so it's not, it resolves into safe components. That is where it's going. For the removal, that is the case. And so the challenge with that is that patients have to have multiple surgeries. So you have a surgery to receive the implant and you have another surgery to take it out. So the progress is where we are trying to design the material such that after that period, it resolves without any negative bioaccumulations in the body. It just gets out of the body.
There
Yes.
is another question for you from Samuel. How can AI be applied to help reduce the need for animal testing? And do you think it can completely replace human testing? Uh, this is
Okay.
this is something you should also add to where the field is going. Go ahead.
Yeah. So, in terms of reducing the need for animal testing, so you see, like, the case of material modification. Like I said, after one stage, which is the in vitro, the next is to replace it in the body. Sorry, in the animal testing. So how AI comes in is that we know what we have gotten from any modification we have done. So that means we understand the structure, we understand the properties. So then we can do machine learning with the physiological states of the animal and make some predictions. So even with what I shared, I did a certain temperature. It doesn't mean that is the best. So AI can help me do multiple experiment and come up with, data that can predict that, okay, you have done 900 degrees Celsius. If you did 1,000, this is what you would get. So instead of each one going into the animal to test, that is how machine learning would reduce it.
All right. Dr. Nutifafa want to ask a question. Please unmute and go ahead.
Thank you, Sarah. Thank you for a wonderful lecture. I would say I'm not in the field, but I can keep one or two things today in terms of biomaterial. I know you also talk about careers. All I know about biomaterial, reading a bit from Osafo are those tissue engineering and how we can go nanoparticle for drug delivery. So today I've heard a few other dimensions. My question was actually related. I was the gentleman who asked a question about AI. So it's just a coincidence. But I'm trying to rephrase it now because I feel like it's not. So it could be repetitive. So if it's repetitive, just answer what you can answer and then just move on. But my point is, I know AI and machine learning can be a good tool, powerful tool for work. But in terms of biomaterial, biomedical research, and especially the example that you just touched on or you gave, and the gentleman also asked a question about what it has to do with animal studies. Will it be because I know mostly AI and ML are very powerful when you have enough data. In some field, they still have difficulty to implement it because there is not enough data to train your models and models on to be because it's a predictive model. So you want them to be more reliable. In this case, so my question now, based on what the question the gentleman asked, do you feel, or based on your experience, do you think that in the field of biomaterial, you have enough data available to train these models on, so that when the next example you gave about temperature, so that when you feed him the AI with, okay, we want to perform a new experiment with the amount of data you have already, the training you have, we have performed this one experiment at 25 degrees Celsius. Can you predict the next result for us? So the question is, do you have enough data to really do that? And then also there's a question I just read, that is computational studies the same as AI and machine learning? That's dicey. I can say no, but they are kind of interrelated. So please, that's my question.
Okay, so thank you for answering the other one for me. Okay, so with even in my group, now that we are doing AI on some of our, experimental data, that challenge of small data is real.
Okay.
Because so there is us in the lab doing so many experiments, then we hand the data over to the machine learning experts, and they are always asking for more. And it feels like it takes a lot even to get what you have given them. So that challenge
Yeah.
is there.
Yeah.
So even this morning, I was like, okay, so is there a way? So the field is growing, is evolving, and all of that. So we can tackle some of the tools along the way and not wait for it to get to an extent. So the question is, I'm answering it in the form of question. If there are any AI experts here, instead of making that challenge of our data is not much an embarrassment, how about we come up with models that are able to work with the little we have? Without that, for now, what I can say is that we really need a lot of data, which we sometimes don't have. So it's also a challenge that needs to be addressed. And that challenge always is an opportunity for other people with other expertise to advance the field.
Yeah,
Yeah, thank
so
you. I thought it's just in my field, so
Mm.
now,
I'm kind of satisfied now. But I think what people can also do, and some people have started in our field, is to create a data bank or a data center where, or people call it repository, but I don't want to call it that because we have repository mostly for literature and stuff like that. But this one is about data. So you have a data center where you can mine. When I say mining, people can actively mine data in the case of kind of importing whatever they have already. But who regulates that? That goes to another realm, right? So I don't want to enter that realm. So if you have that data center where your data that you have in the lab that people are calling inadequate for training the model can be dumped there, and any other person around the world that is doing the same, research can dump them there, then it helps the AI model people to really have enough data on hand. But that's also something it's things are going fast than
Yeah.
policies and stuff. So we don't want to enter that. So but thank you for your reply. I appreciate
all right. So we have some 3 minutes to go. There is a professor in WPI, Massachusetts, in the US, who is specializing in developing machine learning algorithms for small data because they've had that issue. And he's gotten modules that can accurately, to some extent, classify stages of cancer from images. So I think they've published two papers for the work. So that is what. All right. So that is also ongoing. So people are already beginning to do that. Okay, let me just mute. All right. So people are already doing that. People are now developing modules that can use small data to do to classify materials and all that. So the question you asked was justifiable. Instead of saying we should have more data, can we develop algorithms that can still be predictive with small data? And it's already going on. And just classifying stages of cancer from images is something that is already being done with small amount of data. All right, Bene, please, if you ask a question, please go ahead.
Yeah, even though I said my question in the chat, but I just want to find out because Dr. Nancy talked about the advancement of material science with biomaterials in helping to replace organs in the body. So I just want to know what is the level now of material science as it relates to biomaterials in the replacement of organs in the body.
Okay, thank you. So it's at the level where when it comes to printing the organ, that is it's not the difficult part. You know, the organ is filled with network of veins and all of that to allow the flow of blood, which is called vascularization. So for instance, even with the implants, when we put it there is always the movements with the natural environment, veins that blood are moving to. So a lot more research is going into how to be able to make these and it's a network of so many things to allow the flow of blood. So that's an area that people are going into now to design all these materials that we can be able to have those network where blood can flow to and fro in the body. So that is quite a fairly new area, and it's a very interesting one. And I think to begin with, for instance, with the vascularization, I was looking at how to incorporate something like that in my study, and I found out that there are different approaches. So you can start from a very simple approach where there is something called angiogenesis, where the tissues are able to, give you signals that when you use that material, it can be able to form those connective tissues. So those are very, very simple experiments that are done in labs that can maybe have an incubator and all to do the simple ones before advancing them to a stage where you can actually do the real studies with actual endothelial cells to observe these ones.
All right.
Thank you very much, madam.
Somebody's hand was up, but the person has lowered it. If you still want to ask your question, we'll take that as the last question and then wrap up this evening. This is the Young African Scientists platform. We now have a LinkedIn page, and what I would do is I'll send a link to everyone, and so that you can join the Young African Scientists LinkedIn page. I'm going to drop the we've been in existence for a while, but we just had that group, that page done because we interact on other platforms. So the LinkedIn platform is done. I'm dropping the link in the chat. Please go and follow us, and we'll bring you a lot of content like this. We are going to have next month. We are also going to have a series of presentations. So I want you to come back for more. Be like Oliver, come back for more. And we are going to also have what we call the panel, where if you're doing any work in any part of Africa or the world and you want seasoned professors and seasoned researchers to go through your work and to suggest some objectives or some ideas for you, we are going to float the registration form very soon, and we'll register people that want to do that. So you come, you give us a 15 minutes presentation, and then based on that, we'll have seasoned professors that will just comment on your work and help you shape the work in some way that you want. So your presentation should be at a very high level so that you can protect some of the things that you want to protect. But at this level, it will just be ideas, not necessarily results and discussion. So whatever that you want to bring along, you will get people to help you shape the idea. Towards November, we are also going to have a conference, an online conference, and we are going to deal with 3 thematic areas. We'll give you more information about this as the ideas and the plans come into fruitfulness. So we'll come back to you and we'll give you all of those information. But for now, thank you very much for coming around. It's a pleasure to have you around. If you have not registered, I'm putting the link once more. You can just tap on it and register so that we can get your information and send you the certificate. Dr. Sarah, as we wrap up, your last words.
Okay. Since I believe this will go far, but most of us here have our roots and our connections to Africa. Mostly what I like to highlight is that challenges are opportunities, and even opportunities always would present challenges. And that is how any field advances and grow. So whether it's a challenge you have or it's an opportunity that you are presented with, you try and make do with the most. And for you to be able to contribute, you should have something. So what every one of us would have access to is the understanding of the science that is not hidden from us all. So that is my message to us all. When we have understanding of whatever field that we are working in, we'll be able to make an impact. Thank you so much for your time.
All right, loud and clear. The message is clear. No matter where you are in Africa, doing basic experiments is something that you can do. And we have a lecture. I don't know when I will give that lecture, but there is a lecture on how to design simple experiments. No matter where you are, whatever tools that you have, how do you use those tools to do very interesting work and work that you can even publish. For instance, you can use just a hardness tester to actually publish a very good in a very good journal. So thank you very much, everyone, for coming around. It has been good to have you. This is a recorded meeting, and we are going to post the recording in our groups. There is a WhatsApp group here, and I'll send the link around if you're interested to join the Young African Scientists WhatsApp group. You can send the email that contacted you a message, I want to join the WhatsApp group so that I can get all the information that floats around on a weekly basis as well. Thank you very much, everyone, for coming around. It's good having Nutifa, Farnesta, and all the other guys around as well. God bless you, Dr. Sarah, for the insightful presentation. We are proud of you, and thank you very much for the hard work.
Thank you.
All right. Bye-bye, everyone, and see you in the next 2 weeks. Bye.